When it comes to identifying the bacteria present, what we want to do is determine what genus it is: Escherichia, Staphylococcus, Streptococcus. We want to determine that as quickly as possible. So what we're going to see over this section 3 of chapter 15 are methods that are tried and true that, well, some are going to be faster than others. One of the first things that an ID lab is going to do, remember ID labs, infectious diseases labs, once you take the swab, the clean catch from a urine sample, you've resected, taken a section out of infected tissue, you send it off. It's the ID lab that is going to attempt to identify what's causing the infection. One of the things they're going to do, first steps, gram stain. Let's see what's there. This is going to be, down and dirty, really. There may be a lot of bacteria present. There may be very few bacteria present, depending on what the sample is, depending upon where it was taken from. Will this give them a definitive, this is what it is? No. Remember, it's a visualization of the shape, arrangement. Are they clustered? Are they in chains? And then gram positive, gram negative. Now, with those things, you can begin to narrow down the potential antibiotics that could be used. Another portion of this phenotypic methods is going to be looking at, the metabolism. Can this bacteria grow on this media? Can this bacteria grow on this media and use this special whatever added to it? You look at the dichotomous key right here. Gram positive, gram negative, cocci, bacilli. And as you go through each of the different branches, those branches keep branching and going and going. You look, cocci or bacilli. Okay, you're looking at a gram stain, it's cocci. Gram positive, or is it gram negative? If it's gram positive, catalase negative. If it's catalase negative and it's a gram positive cocci, pretty much it's going to be some form of a streptococcus. It's a gram positive cocci that's catalase positive. Then you look whether or not it's aerobic, facultative anaerobe, micrococcus, staphylococcus. And you just keep going through looking at these different branchings. Is it A or is it B? Is it C or is it D? Yes, no, yes, no. By doing this, turns out pretty much all have their own unique pattern of yeses and nos. To be a yes means it has to have the genes present to make some form of a protein or some form of a transport protein, has to have the ability. Okay, to have these abilities, 1, 2, 3, 4 abilities, means it has to have 1, 2, 3, 4 genes or groups of genes to be able to do that. And it's the different collection of genes is what's going to give you the different species. The problem is using the different augers, the different broths, going through testing, the abilities, can it use this sugar? Can it ferment? Things like that. It takes time. Okay, you're gonna have to culture the bacteria for a while. So it's going to take hours to, few hours to up to 12 hours. Gram stain, looking at it that way, takes a few minutes. So this is why in a clinical setting, you'll see the doctor, the nurse practitioner, the PA will prescribe a certain antibiotic because there's an infection and they can't wait. And then 12, 24 hours later, they will change the prescription, either change the dosage or completely change which antibiotic is being used. As they get information back from the infectious diseases lab, they will go through and they will modify current treatments.